Welcome to our lecture on sleep, in which we will discuss polysomnography and the circadian rhythm. We will very briefly look at sleep physiology, and finally we will conclude by talking about what the functions of sleep might be, and we will give an example of sleep disorders. EEG is widely used in sleep research, also called polysomnography. The first EEG recordings were obtained by Richard Caden from the brain surface of animal models, and subsequently Hans Berger managed to record the first electrical signal from the human scalp. And this technological advancement marked the beginning of sleep research. Other pioneers of sleep research were Eugene Aserinsky and Nathaniel Kleitman. Both of them, for example, observed that when people sleep and reach a specific sleep stage called rapid eye movement, or REM, they make very abrupt eye movements and also report having vivid dreams. Modern polysomnography can entail more than EEG. Additional measurements can be electromyography, electrooculogram, pulse oximetry, respiration, heart rate, and many more. Now let's look at sleep stages and the hypnogram. The hypnogram is a graph that shows the progression of sleep stages over a typical night. And we see the alternation between rapid eye movement and non-rapid eye movement sleep stages. And they alternate about 90 minutes. An individual typically progresses from the wake stage into the first non-REM sleep stage, N1, which is a light type of sleep, then moves towards progressively deeper non-REM sleep stages, N2 and N3, then circles back to a lighter non-REM sleep before reaching the first REM sleep. And as the night progresses, we observe that people typically spend less time in the deepest sleep stages and more time in the REM stage. Now, if we look at typical EOG, EEG, and EMG signals during wakefulness and sleep stages, we observe noticeable characteristics. During wakefulness, for example, we see voluntary eye movements, we see fast low amplitude EEG signals, and the EMG reflects variable muscle tone. During N1, we observe a slowing of EEG frequencies and of eye movements and a decreased muscle activity. During N2, we observe now sleep spindles, which are bursts of 12 to 14 Hertz, and high voltage slow waves, also called K-complexes. During N3, we observe high voltage slow waves. And finally, during REM sleep, we see that the EEG signal looks very similar to that recorded during wakefulness. We observe the rapid eye movements characteristic of this stage, but we also observe very decreased muscle activity. During stages N2 and 3, people are generally unconscious of the world around them, so they do not typically respond to light stimuli. Across all stages of non-REM sleep, eye movements are absent. The muscle tone is low, and the breathing is slow and regular. Body temperature also falls, and it continues to fall as we go towards the REM stage. During REM sleep, muscle tone is very weak due to the inhibition of motor neurons, but this paralysis, of course, does not affect the neurons that support respiration, eye movements, and sphincter control, but it is sufficient to prevent us from enacting our dreams. Body temperature falls even further. We observe large variability in heart rate and blood pressure, and people report having vivid dreams. The circadian rhythm reflects the alternation of sleep and wake cycles over typically a period of 24 hours. A pioneer of the study of the circadian rhythm was Nathaniel Clayton, who, together with his student Bruce Richardson, in 1938 descended into the Mammoth Cave in Kentucky and spent 32 days. They chose this particular setup due to the lack of natural light, the absence of sound, and the constant temperature. And the purpose of their study was generally to see if they can entrain their circadian rhythm, so to see how flexible circadian rhythms are. Now, circadian rhythms are intrinsically connected with homeostatic regulation, and we can see that body temperature, growth hormone secretion, and cortisol secretion vary across a typical circadian rhythm span. So the core body temperature and blood levels of growth hormone and cortisol show this rhythmic pattern that is entrained to this rhythmicity of the circadian rhythm over the 24 hours. Specifically, we observe that in the early evening, our body temperature begins to decrease, but growth hormone begins to increase. The level of cortisol, which is the stress hormone, begins to increase towards the morning and remains elevated for a few hours. Sleep also varies across our lifespan. So if we look at how many hours we spend sleeping in a day, we can see that from birth towards our old age, we sleep less and less, and we spend more and more time awake. Interestingly, sleep stages and sleep-wake cycles also vary. So if we look at the graph on the right side, we see, for example, that REM sleep is most predominant in newborns. And this has been hypothesized to help brain development. the structures that support the sleep-wake cycle and that act as controls for inducing wakefulness or promoting sleep are part of a very complex system. And we can see that these structures can be involved in arousal, so they can promote wakefulness. They can promote sleep. They can induce a REM sleep stage, or can be involved in circadian regulation. However, there are brain areas that perform many of these roles. At the center of this complex story, however, lies the suprachiasmatic nucleus. We consider it to be the central pacemaker. It is located in the basal hypothalamus on either side of the third ventricle, and it contains two nuclei. Each nucleus contains in turn around 10,000 neurons, which are individually specialized to be able to act as circadian oscillators. And so they show a high activity, a high firing rate during the day, and low activity during the night. The main hormone that induces sleep is melatonin, colloquially called the hormone of darkness. And this hormone is synthesized by the pineal gland and is released in response to darkness. So why do we sleep? Well, restorative theories hypothesize that sleep plays a variety of functions, mainly focused on repairing and consolidating our immune response and consolidating our memories. Perhaps sleep might also support detoxifying processes. With respect to REM sleep, specifically, being characterized by intense neuronal activity and energy expenditure, and because we observe it being extensively present in newborns, it may have a role early in life in establishing the neuronal connections that have been genetically programmed. So what happens when sleep regulation goes wrong? One example is narcolepsy. Narcolepsy is a central nervous system hypersomnia disorder, meaning it is characterized by increased sleepiness, by cataplexy, which reflects sudden loss of muscle tone, as well as sleep paralysis. And it also can be characterized by fragmented sleep and insomnia.